Okay, so what we're going to do right now is talk about chapter 11, and we're going to discuss the forms of cell communication, how cells of multicellular organisms are going to communicate with one another. So um, when we look at multicellular organisms, such as ourselves, we consist of trillions of cells. And so these cells have to coordinate some sort of a response. They have to be able to kind of tell each other what to do and respond to the surroundings and things like that. And what this communication enables the organism to do really is to kind of coordinate the different activities of all of these cells and kind of contribute to the overall livelihood, vitality of that particular organism. So the way in which this really occurs is through signal transduction pathways. So there's three main stages to signal transduction. You have reception, transduction, and response. And with reception, typically what's happening is you're going to have the signal from the outside of the cell being received either by a membrane receptor or by an intracellular receptor, just depending on the form of the signal molecule. For instance, if it's water soluble, then it has to bind to a protein on the surface of the cell because it can't pass through the hydrophobic interior of the membrane. And if it's fat soluble, then it can get into the membrane and then binds with an intracellular receptor and then causes the cellular response. But in the case really that we're going to focus on really in this presentation is just those water soluble types of signal molecules that are going to bind to the surface proteins. So the reception, when you have that protein on the surface of the cell, what it's going to do is it's going to receive the signal. And then the transduction aspect of this, step 2 here, this is where you have a change in a shape of the protein structure in some way that's going to actually kind of kickstart a series of events that's going to lead to changes within the cell, either a change in gene expression or a change in the activity of the cell, something along those lines. And that's what the actual response is going to be. Usually you're going to, change the gene expression in some way, shape, or form. So the way in which this happens, if you kind of look here, what we have, here's your signaling molecule. And in this case, this is like the ligand. We can call it the ligand. That's, generally speaking, the smaller molecule that's going to bind to a larger molecule and usually result in some sort of a change. So here's this purple thing here. This is your intracellular or the intramembrane receptor. It's kind of embedded. It's one of those plasma membrane proteins that we talked about earlier in the year. And so you have reception of the signal, and then this is going to kickstart a series of events where the relay molecules are going to be involved, and that's the signal transduction. And then all of that is going to kind of coordinate a cellular response. And then once the signaling molecule goes away, ideally all of this is going to shut off. Okay, so the local signaling now, you can have local signaling between two neighboring cells. And a lot of times what you'll have is either a gap junction or a plasmodesmata where these molecules produced by one cell can kind of pass back and forth in between the cells and so forth. Cells might be joined together by desmosomes or certain types of intracellular junctions that'll hold them together. Here you see a plant cell with the plasmodesmata. Again, what is being produced by one cell can pass back and forth. So that's a form of local signaling. And then another form of local signaling can be either like a juxtacrine signaling or a paracrine. So if you think of juxtacrine like being juxtaposed or on top of, juxtacrine signaling is signaling through the touching. So you can see here you've got one cell that's actually physically in contact with or touching another cell. And that touching can kickstart a series of events that's going to lead to a response in that other cell. Now with paracrine signaling, para means beside. So in paracrine signaling here, you can see you've got cells that are immediately adjacent to one another, right next to or on the side of one another, and they're releasing substances that are having an impact on the other cells. So for instance, you have cells in your immune system really that work this way, where, for instance, like a helper T cell might be releasing some substances that are going to kind of get cytotoxic T cells going or B cells going or something along those lines. So you can see they're releasing chemicals into the environment that are having an impact on the neighboring cells. So that would be paracrine signaling. Another kind of specialized type of localized signaling that we would see in animal cells would be synaptic signaling. So you can see here, this cell, this would be the presynaptic cell, and this is the postsynaptic cell, which you can see the nucleus in. And so you'll get an action potential, which is electrical activity. That's like the way in which the cells are going to kind of communicate. These nerve cells will communicate with one another. And so the electrical activity, the action potential, will travel down the axon. And when it gets to the axon terminus, the end there, usually that electrical activity will stimulate like an influx of calcium or something like that. And these calcium ions will stimulate the exocytosis or the release of these neurotransmitters into the synapse. And once they get into the synapse, what they're going to do is they're going to act like the ligand now, and they're going to bind to, which you can't really see in this diagram here, but they bind to receptor proteins on this postsynaptic cell. And what they basically are doing is they're allowing the ions to come in, and then the generation of an action potential in this postsynaptic cell here, and then the communication will continue onward, okay? Now, plants and animals both here, they'll use like a form of long distance signaling. This is like endocrine signaling. So you will have some sort of an endocrine cell or endocrine tissue that's going to release the hormones into the bloodstream. And then once those get into the bloodstream, they're going to travel through the blood and then interact with their target organs. So when they get into the blood, they're going to, have basically contact with every type of cell in your body. But there's only certain types of cells in your body that are going to be responsive to some of these hormones. Some are responsive to more than others. But if you figure something like antidiuretic hormone, when you get really thirsty, your posterior pituitary will release the antidiuretic hormone, and that's going to, be coursing through the blood all in your body. But it's really only going to have a dramatic impact on the epithelial cells in your kidney, and that's going to help stimulate the reabsorption of water because and the concentration of urine, because you're thirsty and you don't want to be getting rid of this excess water. So that would be an example of long distance signaling where you're going to target specific cells. Something that might be a little bit more general would be like the release of insulin, and you're going to have cells, all through your body that are going to be able to respond to the insulin, so you can take up the sugar out of your bloodstream, and so on and so forth. So there's lots of different examples, but that's an example of the long distance signaling. So the main question then is, so how does this all happen? And the main way that generally has two options is you have this receptor molecule that's going to be on the surface of the cell, like I mentioned earlier, and it's going to receive the signal and the information that it carries and transmit it throughout the cell, cause the response. Or you might have, again, like we said earlier, if you have a lipid-soluble signaling molecule, it can diffuse into the cell and bind with an intracellular receptor and then cause a response that way. And then once you get that taking place, that's when that signal transduction is going to take place, and then ultimately end up with a response of the cell. Okay, so if we look at this in a little bit more detail, here is the reception phase. So what's going on here is we've got this target cell, and here's this orange molecule here is going to bind to the receptor. In this case, this is a G protein coupled receptor. And so it binds to this receptor, sends a signal to another protein embedded in the membrane, and then that's going to kickstart the cellular response. Whereas if you have a lipid soluble hormone here or signal, it can diffuse through the plasma membrane, bind to a signal receptor or a hormone receptor inside of the cell, and then you can see that it can cause this cellular response. In this particular case, happens to be kickstarting transcription and translation, manufacturing of a protein, and so on and so forth. Okay, so with reception, this is where we're going to receive the signal on the target cell. And this is very similar to an enzyme substrate reaction, whereas it's, there's some specificity involved, as I mentioned earlier. That signal, again, is the ligand, and binding is generally going to cause a change in shape of that protein, which is going to then therefore lead to some sort of an activation of that. So as mentioned earlier, the plasma membrane receptors, these are usually for water soluble types of hormones. So these are going to bind to the protein on the surface of the cell and then transmit that signal to the inside of the cell. And the three major types of plasma membrane receptors that we need to be concerned with are G protein coupled receptors, receptor tyrosine kinases, and ion channel receptors. Sometimes you see the G protein coupled receptors as being GPCRs, and sometimes you see the receptor tyrosine kinases as referred to as RTKs. So if you're doing some research on this on the internet or looking through your book, you might just see those abbreviations occasionally. Okay. So G protein coupled receptors are very widespread, very diverse in their functions in the animal kingdom and so forth. And that's why we, learn a lot about them. They're implicated in a lot of diseases, embryonic development and so forth, as it says here. And then one example, if you look involves smell. So these are your epithelial cells in your nose. And then these are the nerve endings that lead to the olfactory bulb, the thing that's going to process your sense of smell. And so you can see the ends of these nerve cells, the dendrites. These are going to kind of branch out into the mucous membrane that is on all surfaces of like our nose and throat and sinus cavities and things like that. But particularly in our nose, this is where these kind of little strings of nerve fiber are going to be embedded in this mucous membrane. And when we encounter some sort of an odorant molecule, it's going to bind to these G protein coupled receptors that are on the surfaces of these cells. So if we blow up these little extensions here, here's like the cell membrane, and this thing labeled R, this would be like the G protein coupled receptor. And so that GPCR is going to bind this odorant molecule, and it's going to relay that information to adenylyl cyclase and other protein embedded in the membrane. And then that's going to kickstart a series of events, intracellular events, this signal cascade. And that signal transduction is going to lead to the generation of an action potential that's going to get transferred along these neurons to the olfactory bulb so that we can process that sense of smell. So that's just an example involving odors. Another thing, pheromones, hormones, neurotransmitters, certain disease toxins, these things all play a role in binding with G protein coupled receptors. So we learned in like chapter 5, when we were talking about macromolecules, about alpha helices, beta pleated sheets, different formations of the protein structure. Well, G proteins all have 7 alpha helices that are kind of embedded in that membrane. And you've got a binding site for the signal, and then you've got a G protein interaction site kind of on the inside there. And this is what's going to bind with those things that we were just talking about. So if you look at this diagram, here's your GPCR. And here's the G protein on the inside of the cell. And so it's going to interact with an enzyme that's going to kickstart this pathway. So what happens is when the receptor gets activated, it activates this G protein. So it takes it from a low energy state where GDP is bound and binds it with GTP, a high energy compound, and that activates it. And what happens is it slides over and activates this enzyme. And as soon as it releases that high energy phosphate, of course, the G protein then slides back into a low energy state. But what you've done is you've activated this enzyme here, this adenylyl cyclase usually enzyme. And this enzyme now is going to coordinate a response, a cellular response. And the more signaling molecules you have, the heavier the response is. But as soon as the signaling molecule goes away, the G protein doesn't get activated again. And then like activity, the intracellular activity is going to shut down. Okay. So if you look here, these two diagrams are kind of showing this. If we focus on the one on the left here, this red or maroon kind of structure, this is binding to the G protein or the G protein receptor, excuse me. And then, so you can see here it comes in, it binds, it activates the G protein with the GTP, slides over and then activates this enzyme, which is going to coordinate the intracellular response. And here you can just see on the right in this figure that's moving a lot quicker, this is the signal is bound here. So you can see as the signal is bound, this G protein is just going to continually slide back and forth and result in an increase in the cellular activity on the inside. So as soon as the G protein activates the enzyme, it loses the energy, so to speak, and it moves to a low energy configuration. As long as the signal's there, it re-ups with a GTP molecule, gets heavily activated, and then slides over. So you can see how it slides back and forth between an activated and an inactivated state. Okay, so the G protein coupled receptor is just kind of slowing it down in looking at this term in terms of like a fight or flight response. Like when you get that adrenaline rush, you're scared and you've got to kind of flee the situation or whatever. So you get this epinephrine release. This binds to the GPCR, activates the G protein, and then the adenylyl cyclase is going to kickstart this second messenger, cyclic AMP in this case. This second messenger is going to have an impact on protein kinase A, PKA it's sometimes called. So here is your signal cascade. The activated adenylyl cyclase gets the second messenger, cyclic AMP going, which kickstarts the protein kinase, which then leads to a number of things. In the case of this like adrenaline rush, we're going to stop the synthesis of glycogen. We don't want to store glucose. What we want to do is break it down. And the other part of it here is, again, promoting the glycogen breakdown. So when we look at this, here's the signal cascade. Here's where one molecule or a small amount of signaling molecule can have a massive impact on the outcome of what's going on. So for instance, here's your one molecule of epinephrine that's been released. And obviously there's a lot more than one molecule that gets released, but this is just showing you the kind of magnification of this process. One molecule gets released right here. And then what happens is your signal transduction pathway that activates the G protein. Maybe one molecule activates 100 molecules of this G protein, which activates 100 adenylyl cyclases, which are going to in turn get 10,000 or 10 to the fourth cyclic AMPs going. And then those are going to activate another 10 to the fourth protein kinase A molecules, which are going to lead to the activation of, and then the release ultimately of 10 to the eighth molecules of glucose. So you can see this signaling pathway as we continue on. We're going to activate glycogen breakdown, the release of the glucose molecules so that it can activate the cellular respiration pathways, give us that quick ATP energy so that we can power our muscles to, flee the predator or the situation that stimulated the release of the epinephrine in the beginning. Okay, so the other thing about G protein coupled receptors is they're involved in numerous kinds of diseases. So for instance, one of the ones that we've learned about or talked about briefly was cholera. And then the other one is pertussis, which is whooping cough. And this interference of the G protein from these toxins in the way that they're normally supposed to function is what causes the symptoms of the disease, these massive coughing attacks, a lot of diarrhea, which ultimately results in dehydration and death and things like that. So if you look at the cholera toxin, what happens with cholera toxin, it gets brought into the cell through endocytosis, it gets processed, and then when it's sent back to the cell membrane, this is where it causes a lot of problems, okay? And what it does is it increases the rate or the activity of the enzyme, which increases the adenylyl cyclase, or the receptor really, it gets overactivated. And then the adenylyl cyclase is highly activated. And then this leads to a cascade of events in overactive form of these events, which ultimately leads to these CFTR proteins. These are chloride channels that leads to an efflux of chloride. So in other words, it's normally in a delicate balance of what's going on, but this toxin interferes with this, and then you're overstimulating the efflux of the chloride from the chloride channels, or through the chloride channels rather. And what happens is the efflux of the chloride from the tissues that they're supposed to be in into the lumen of the intestine, like where the food moves through, into there. When the chloride goes in there, that throws off the whole balance. And it leads to the loss of water into the intestinal lumen, sodium ions, potassium ions, bicarbonate ions, and so forth. So now you've thrown off the entire water balance. And what this leads to is rapid dehydration and electrolyte loss because of the massive diarrhea, and like 2 liters an hour, that is very quickly will result in death. So within 24 hours, you can go from being totally healthy and normal to this bacterium getting into your system and then causing this death if it's not treated through dehydration and the electrolyte loss. Okay, so that's how the G protein is implicated in cholera. And so the way it works with pertussis is very similar, only the opposite way, in that when the toxin gets in there, a different subunit, a different G part of the G protein subunit is actually going to become inactivated. And now what it can't do is, you can't inhibit this, you can't shut the process down. So there's a way to turn it on in either case, and there's a way to shut it off. And in this case, you can't shut it off. So now, since you don't have a way to shut off that adenylyl cyclase molecule, it's still going to be in overdrive. And in this case, what it's going to cause are a bunch of symptoms of the disease of the whooping cough. Okay, so pulmonary hypertension, it's like a lung disorder. You're going to have hypoglycemia, the fever, and all of those sorts of things. So if you look at the receptors here, In the case of the cholera on the left here, what we're doing is we have an overactive G protein, okay, because now we're in overdrive, but this particular subunit of the G protein is causing endless stimulation of adenylyl cyclase, and therefore the symptoms of cholera. And on the right, what we have is pertussis, which is going to work similar fashion, but now we can't inhibit the adenylyl cyclase once it gets going. So a different subunit is activated, or rather inactivated, and therefore it can't shut the process off. So adenylyl cyclase still remains in overdrive. So here it's stimulated in the positive way, where it's just continually go, go, go. And on the right here, it's going and we can't shut it off. Okay, so that's how G proteins are kind of working for the purposes here of G protein coupled receptors and signal transduction, and how it's supposed to work, how it does work in some instances, and then what happens if we have like a disease of some sort, or a toxin and an interference with it. Okay, so the next thing we're going to start with in the next video will be the receptor tyrosine kinases. All right, so if you have any questions on any of the video that you just watched, of course, like always, just make sure you write them down, and then we'll talk about them in class. Okay. Talk to you soon.